All right, class. So I hope I was detailed enough in my drawings and you have a good understanding of the countercurrent multiplier mechanism and also the countercurrent exchange mechanism that takes place in the vasa recta. Now we will quickly, very quickly go over the PowerPoints to make sure you understand it 100%. So if you look at the diagram here, we are looking into the ascending limb of the loop of Henle. This is the lumen side, this is the tubular lumen side, and this is the interstitial space. And the cell that you, the orange, not orange, the tan colored cell that you see in between, these are the bad boys I drew out. Remember the bad boys? All right. So remember the first thing you have to remember is that the ascending limb of the loop is impermeable to water. So it does not allow water to move, right? So from the filtrate, do you see this transporter? From the filtrate, sodium is transported into the cell. So sodium comes inside the cell. Along with sodium, there are two chloride ions that come in, and also potassium ion comes in. Some of the potassium ions that come in actually moves back into the filtrate. That's okay. And then we cannot have too much sodium chloride or potassium in the cell, right? So sodium from the cell goes out into the interstitial space by this sodium-potassium pump. And in return, 2 potassium comes in. By some special chloride channels, potassium and chloride also leaves the cell. Therefore, what do we end up with? We do see that the interstitial space gets a lot of solutes that get a lot of solutes from the filtrate, right? That's what we see here. Okay. Now, so this is the ascending limb, right? This is the ascending limb. This is the descending limb. All right. So as we mentioned before, the ascending limb pumps out salt. Now look at this. Look at the filtrate that enters the descending limb. Ooh, it enters. It's at 300 milliosmoles. But as it enters, it finds the surrounding very hypertonic. So the filtrate at this point feels very hypertonic. Very hypotonic. The filtrate is super hypotonic, right? So what will it do? The filtrate, what will it do? The filtrate, being hypotonic, will let water move out. And remember, the descending limb is permeable to water only water. So as water moves out, the filtrate becomes more and more concentrated. And that is a good thing. The more concentrated the filtrate becomes, the more salt will be excreted out, or the more salt will be pumped out from the ascending limb. Okay, and the water that comes out actually enters capillaries and gets back to our circulatory system. All right. So this is, I have explained this. And so remember one thing, the countercurrent multiplication mechanism, it actually establishes a positive feedback mechanism between the two portions of the loop. So the idea is the more salt pumped out by the ascending limb, the more water will come out of the descending limb. So more salt out from the ascending, more water out from the descending. So that's a positive feedback mechanism that's taking place here. All right. So this is just a summary slide. I will leave this for you to study on your own. And I'm not explaining these two because I have talked enough about the countercurrent multiplier system. Now moving on to countercurrent exchange system that takes place in the vasa recta. Remember, vasa recta is a blood capillary that runs along the loop of Henle. Vasa recta also has a descending portion and an ascending portion, right? So And this vasa recta actually help create the countercurrent system because they take in a lot of salt in the descending region. So let's look into the descending region. As you can see, the black arrows represent intake of salt. Intake of salt. But they lose them again in the ascending region. Look into the ascending region. Are we losing salt here from the ascending? Yes, look at the black arrows. We are losing salt here. And this mechanism helps to keep the gradient, helps to maintain the gradient of the medulla. Okay. So the high salt concentration in the beginning of the ascending region pulls water. So that here, at this here, the salt concentration is high, right? So that pulls in water from the interstitial space into the blood capillary. And this actually all together, everything together keeps the salt concentration in the interstitial space high. All right, that's it. And I'll start with a new video.